Mitochondria and chloroplast, they are energy converting organelles. As I mentioned earlier, they are not the members of endomembrane system. So very different. So mitochondria and chloroplast change energy from one form to another. Mitochondria are the site of cellular respiration, a metabolic process that uses oxygen to generate ATP. That's the direct biological energy form, ATP. And chloroplasts found in plants and algae are the sites of photosynthesis to convert sunlight energy into our food. Those two organelles, like mitochondrion and chloroplast, they are very funny. They are very exceptional. It's funny because they have those two organelles. Exceptionally, they have their own DNA and they have their own ribosome, very different from in cytoplasm. So they have their own DNA, they have their own ribosome. That means they produce their own protein, and sometimes like the living organism, those mitochondria and chloroplasts, sometimes they go through division. So one mitochondrion divide into two, and two become four, four become eight, chloroplast same. So they go through something like cell division, like the living organism. So not the cell division we call, but they go through division to increase their number. So that's why they are very funny. So they are very exceptional. So mitochondria and chloroplasts have similarity with bacteria, living bacteria, independent organism. They enveloped by a double membrane, so it is surrounded by double membrane, and contain free ribosomes and circular DNA molecule. They have their own DNA, and they grow and reproduce somewhat independently inside the cells. So this similarity leads to the endosymbiont theory. The endosymbiont theory suggests that an early ancestor of eukaryotes engulfed on oxygen using non-photosynthetic prokaryotic cell. So some cells, they eat up those mitochondria, eat up, or maybe some mitochondria invade into some foreign eukaryotic cell inside. And somehow, by the evolutionary process, they will now become a member inside the cell. Member, like become a organelle inside the cell, something like that. So the engulfed cell formed a relationship with the host cell, becoming an endosymbiont. The endosymbionts evolved into mitochondria. At least one of these cells may have then taken up a photosynthetic prokaryote, which evolved into a chloroplast. So that means, long time ago, maybe mitochondria and chloroplast, they were independent living organisms, but somehow they go inside the eukaryotic cell, and by the evolutionary process, they became organelle inside the eukaryotic cell. Now, they function as a mitochondria and they function as a chloroplast, go through cellular respiration and go through photosynthesis. This diagram showing you the endosymbiont theory. So those mitochondria invade or engulfed by the bigger eukaryotic cell. So somehow they go inside the cell and become organelle. Now, another independent photosynthetic prokaryote, they go into the eukaryotic cell, and by the time passed, it's gonna become a organelle inside the eukaryotic cell. So that's the scientists, they just guess for the origin of those mitochondria and chloroplast, because those mitochondria and chloroplast, they are very funny. They behave very funny. That's why. Mitochondria, for the chemical energy conversion, mitochondria are in nearly all eukaryotic cells. So almost all eukaryotic cells, they have mitochondria inside. They have a smooth outer membrane and an inner membrane folded into cristae. So it is surrounded by double membrane. Outer membrane, it is kind of round and smooth. And inner membrane, it is folded up severely. So folded up structure, one folded up structure we call crista. And more than one, many folded up structure we call cristae. So that's the inner membrane create two compartments, intermembrane space. So intermembrane space means the space between outer membrane and inner membrane, intermembrane space. And inside the inner membrane, we call that the mitochondrial matrix. Some metabolic steps of cellular respiration are catalyzed in mitochondrial matrix. Cristae present a large surface area for enzymes that synthesize ATP. So they making up severely folded up structure to increase up maximum surface area of inner membrane. We're gonna discuss later on in detail in separate chapter. So that's the cristae to secure the maximum surface area of the inner membrane. That's why. For the maximal production of ATP, that's why. This picture showing you the structure of mitochondrion. So it is surrounded by outer membrane, smooth outer membrane, and inner membrane folded up severely, making up structure of crista or cristae. And inside the space of inner membrane we call mitochondrial matrix. And we can find intermembrane space in between outer membrane and inner membrane, intermembrane space. Intermembrane space, it is essential to produce ATP. And inside the mitochondrial matrix, we can find some free ribosomes. And the right-hand side, we can see the image of mitochondrion by TEM. And we can see the network of mitochondria in Euglena. This is one of the members in kingdom Protista, because they have to produce ATP, direct biological energy. Okay, chloroplast. They capture of light energy. They go through photosynthesis. In the whole ecosystem of the earth, the most important and the most miraculous phenomenon is photosynthesis. It is like a magic. I can say it is the greatest magic on the earth. What's the magic? Okay, the greatest magic on the earth, that's the photosynthesis, because sunlight energy, we can see sunlight, but we cannot touch. We can see only as a what? Like the sunlight, just a light. So the concept of the light is not very clear, but anyway, those sunlight, it is converted into our food, our bread, our rice. It converted. Sunlight energy is magically converted into our food by photosynthesis, by the magical organelle of chloroplast. So that's why I call that's the greatest magic. That's the wonderful thing. Okay, anyway, that chloroplast contain the green pigment chlorophyll. Chlorophyll, we call it, as well as enzymes and other molecules that function in photosynthesis. So, in scientific context, we call sunlight energy is converted into chemical energy form. But chemical energy form, we call chemical energy, but it is in just easy words, it is our food. It turned into our food. So chloroplasts are found in leaves and other green organs of plants and in algae, protist algae. Inside the plant cell, we can find some plant cell, it contains many chloroplasts. So that's the overall structure of chloroplast. And right hand side we can see some different shape of chloroplast on algal cell, protist algal cell. So as I mentioned, it's kind of funny. For the mitochondria and chloroplast, they have their own DNA and they have their own ribosome. And it is surrounded by double membrane, outer membrane, and inner membrane. Actually, those outer membrane and inner membrane, they don't have any color. It is clear membrane because it helps to penetrate the sunlight energy into, inside the chloroplast. That's why. And the colored one, we have another additional membrane system we call this a thylakoid or a thylakoid membrane. So those thylakoid membrane, they contain green colored chlorophyll. So that's why it looks green colored. And those thylakoid membrane, we can find the coin stack things, we call this the granum. So they shape up those kind of coin stack shape, granum, and many grana. And those thylakoid membrane, it is surrounded by thick fluid we call that the stroma. Yeah, stroma. The stroma. So stroma, that's the actual site to produce sugar. And right hand side, we can see TEM image of chloroplast. So as we just discussed, chloroplast structure include thylakoid, that's the membranous sacs, and stacked to form a granum and many grana. And stroma, that's the internal fluid, the liquid form stroma. The chloroplast is one of a group of plant organelle called plastids. Okay, so plastids, sometimes they can change. So in seasonal change, so when summertime change into fall, autumn, then what? Those chloroplasts, that their color change from green color into orange color or the yellow color something, then we call that the chromoplast. 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 And when chloroplasts, they go through photosynthesis and stimulate the production of starch, and they stack up a lot of starch molecule, then what? Then color gonna get changed from the green color into white color or colorless. Then we call amyloplast. So those chromoplast, amyloplast, or chloroplast, they can change, they can interchange. So all those, the organelle, we call, collectively we call plastids. Eukaryotic cells, they have some empty space. However, those empty space, it is not empty actually. It is filled with extensive network of small protein fiber network. And those protein fiber network we call, collectively we call cytoskeleton. So the cytoskeleton is a network of fibers that organizes structures and activities in the cell. The cytoskeleton is a network of fiber extending throughout the cytoplasm. So no empty space. It organizes the cell structures and activities, anchoring many organelles. So many organelles, they tied up into cytoskeleton. It is composed of 3 types of molecular structures, so at least 3 types of fiber, protein fiber. The smallest one, not smallest, the biggest one. Biggest one, that's the microtubule. And the smallest one, that's the microfilaments. And in the middle, intermediate one, we call intermediate filaments. Sorry, don't forget, don't get confused. Microtubule, that's the biggest, and microfilament, that's the smallest in their diameter. Roles of the cytoskeleton is for support and motility. What is motility? Motility means moving around, moving from here to there by those cytoskeleton. So the cytoskeleton helps to support the cells and maintain its shape. It interacts with motor protein to produce motility. Motor protein means those special protein, when they spend ATP, they can move around or they can change their shape we call motor protein. It's like a motor, yeah. So like a motor to they have structure to move from one place to another, yeah. Motor protein. Yeah. Inside the cell, vesicles can travel along tracks provided by the cytoskeleton. Yeah. Sometimes cytoskeleton, the protein fiber, it can be a track to ride on. So when they move their location, like a vesicle, they can get along. The cytoskeleton fiber move from one place to another. It's showing you the motor protein. So they ride on microtubule of a fiber for the cytoskeleton, and the vesicle is connected with the motor molecule. So when those motor protein spending ATP, they can act like our feet, yeah, or our hand like that. So when they spending energy, they can move along that microtubule. So they move from one location to another, riding on microtubule like that. So we can see SEM image of the microtubule and the vesicle ride on with the motor protein. As I mentioned, at least three main types of fibers make up the cytoskeleton. First, and the largest one we call microtubule, are the thickest of the three components of the cytoskeleton. And the thinnest one, smallest one in their diameter, that's the microfilaments, also called actin filament, because it is composed of actin protein. That's why sometimes we call just the actin filament. The name is same. Are the thinnest components, and for the intermediate, we call intermediate filaments, are fibers with diameters in a middle range. The table showing you the comparison for those three types of protein fiber. So first one we can see that's the microtubule. Microtubule. How come they call microtubule? The other one is just a filament, something, but it is microtubule. How come? Actually, tubule means is the words comes from tube. Yeah. Tube is what? Tube, that's the cylindrical structure like a pipe. Yeah. Pipe is what? The typical things about pipe or tube, inside is empty. It is same. Yeah. Inside is empty. Only it has outside they rolling up with a protein. They rolling up and like a tube-like structure. That's why the name is microtubule. It's a minute tube, tubule. So it is a microtubule. So that's the first one. And the smallest size one, that's the actin filament or microfilament. So it is composed of round protein actin. So round protein actin, one line and two lines, they twist it up and make up microfilaments. And the third one, that's the intermediate filament. It is composed of not globular protein, not round protein, but it is a long line-shaped protein, keratin protein. So those keratin protein, they roll up and making up something like rope-like structure, and it is very strong, strong and very permanent. So it is the most permanent the filament in cytoskeleton. So I'll give you time to check out by yourself. Sorry, time limited. I had to go on. Sorry, I didn't know the detailed picture. The following, I didn't know that. Sorry for that. Anyway, that's the microtubule. As I mentioned, it is hollow tube, so inside empty. So their size, never mind. So 25 nanometers with something, never mind. And it is composed of round protein tubulin. So alpha and beta tubulin, they comes in all the time. They come along all the time. Alpha, beta, and they roll up, make up a tube-like structure, and become microtubule. And for their function, maintenance of cell shape. So cell shape, because inside a cell they don't have any skeleton, so any bone. So that's why those microtubule, they function as a frame. So maintenance of cell shape and the cell motility. So they involved in cells move around, so as in cilia and flagella. Cilia and flagella, it is like a tail, or it's like a hair. They can move around. So motility, we're using that. So cell, they can move from one place to another. And chromosome movement in cell division. Chromosome movement. So those microtubule, bunch of microtubule, when they go through cell division, they're going to become spindle fiber. Two spindle fiber to catch chromosomes and move from one place to another. They drawing those chromosomes. And the organelle movement. So sometimes when organelle move from one place to another, they using the microtubule like a track. With a motor protein. So they ride on microtubule as a track, and with a motor molecule, motor protein, they ride on and move from one place to another. So that's the function for microtubule. How about microfilaments or actin filaments? So it is composed of round actin protein and two lines of that protein fiber. They intertwined strand of actin. So for their function, that's the maintenance of cell shape and changes in cell shape and muscle contraction. Okay, when we dissect our muscle cell, we can find actin filament and myosin filament. Myosin, M-Y-O-S-I-N, another type of small filaments. So actin filament and myosin filament and actin filament and myosin filament, actin and myosin is something like that. So they alternating each other. So actin, myosin, they stack up like that. And in between those actin and myosin, they have motor protein. So when we contract our muscle, those actin and myosin, they slide each other and they overlap. And when we contract our muscle, when we relax our muscle, they separate out. So they going into each other and they separate each other, something like that. So they slide in and slide out like that with the motor protein. So that's our muscle cell, contract and relax, contract and relax like that. And the other one is cytoplasmic streaming in plant cells. Cytoplasmic streaming means sometimes we call cyclosis. CYCLOSIS. Cyclosis or cytoplasmic streaming. Sometimes using the microfilaments movement, they circulate all the cytoplasm around the central vacuole. They circulate the cytoplasm around the central vacuole. We call cytoplasmic streaming. So by the microfilament, the mass movement, they can go through like that. And the, what is that? Division of animal cell. Yeah? When animal cells go through the cell division, and they produce a two daughter cell, and final stage of the cell division, two daughter cell, they have to separate. Yeah? They have to pinch out. Yeah? Using that microfilaments. Yeah? Microfilament, they making narrower for the connection, and finally they separate two daughter cells each other. By Okay, for intermediate filament, it is produced from fibrous protein coiled into cable structure. So it is a strongest and it is the most permanent. One of the several different proteins, such as keratins, yeah, that's the linear shape protein. Maintenance of a cell shape, tension-bearing element, so because it is strongest, and anchorage of nucleus and certain other organelles. They tied up with intermediate filament, and formation of nuclear lamina. So because it is permanent and strong, it is very good material making up nuclear lamina. So right hand side, we can see a linear shape protein, keratin protein. They twisted up, making up a cable or a rope-like structure. Again, the text coming up. I didn't know that, sorry. Again, yeah, I'm gonna make it short. Okay, microtubules are hollow rod. Never mind, 25 nanometer something. So, and function of microtubules shaping the cell and guiding the movement of organelles because they function as a track, like a railroad for train, and separating chromosome during the cell division. Yeah? So bunch of the microtubule, they making up spindle fiber to move chromosome inside the cell when they go through cell division. Okay, in animal cell, microtubules grow out from a centrosome near the nucleus. And in animal cell, centrosome has a pair of centrioles each with 9 triplets of microtubules arranged in a ring. So two centrioles making up centrosome, and from a centrosome they produce spindle fiber. Actually, those spindle fiber, because those centriole, that's the assembling factory for that microtubule. That's why it is possible. Yeah. So with assembling a microtubule, they can produce a spindle fiber, and those spindle fiber can grow up from a centrosome. The centrosome, it is composed of two centrioles, a pair of centrioles, and that centrioles each other, they crossing each other at right angle, 90 degree each other, make up centrosome. And each centriole, it is composed of 9 triplets of microtubule. So 1, 2, 3 microtubule, they bound each other, make a microtubule triplet. So 1, 2, 3, 4, 5, 6, 7, 8, 9 microtubule triplet, they arranged in ring shape, like a pipe-like structure. That's the what? That's the centriole. So that centriole, that's the, sometimes we call that's the basal body, because that's the basal structure to assemble microtubule like that. That's why that's a very good place to produce spindle fiber from a centrosome. So the central structure, 9 triplet of microtubule, we call that's the basal body.